Hello everyone, and thanks Edmond for the invitation. So I will go a bit more immunology in my 15 minutes presentation, and less high-flying presentation. So I will talk about a cell-based method that to measure antibody that we developed in the lab, and in fact it was a technique that we repurposed from malaria and that we applied for COVID, and we are expanding to other diseases now. So a bit of immunology 101. When you have an antigen encountering antigen-presenting cells, you're going to stimulate T cells and stimulate B cells, and you're going to make memory T cell, memory B cells, and antibodies, or from effector B cells also antibodies. And so that will protect you against an ongoing infection. And if you really make the right T cell and the right antibody level, you could control the infection, and that's the basis for vaccination. When you inject an antigen, you induce a primary response, you boost, you have a secondary response which is faster and higher. And it's both dependent on the T cell and the B cell. So T cells, B cells, and in particular antibody comes at many different flavors. You could neutralize you could neutralize the pathogen. When the antibody recognizes the pathogen, it could be phagocytosed by the phagocytic cells. You could induce antibody-dependent cellular cytotoxicity, especially by NK cells. And you could also induce different other effectors, like the complement, which will lyse pathogen like bacteria, for example. And these different functions come from the FC receptor, the part which is not recognizing the antigen, which are on different flavor, could go from IgG1 to IgGA, IgGE, and so on. And they will engage different FC receptor on different type of cells, inducing different type of reactivities. So you could focus and measure one or the other activity, but usually at the first screen you need to look at what antibody levels we have. I will say the gold standard assay is the ELISA that is used in many labs around the world, and there are many commercial companies sending ELISA for nearly everything. It's good, but sometimes it's a bit long to set up in the lab. You need the antigen and so on. So we decided to take another approach based on the cell expression. We clone a gene of interest into a plasmid which expresses a GFP. We put that in the lentivirus. We transduce cells there. We have the plasmid in a way that the antigen will be expressed at the surface of the cells. And when we come with the secondary antibodies, after we incubate the cells with monoclonal antibodies or serum, we could, in this gate here, detect the percentage of cells which are stained. And we could also control for the number of cells transduced. But because we use lentivirus and we sort the cells, we could have very high expression. And thus, the specificity of the response can be high and very well tested. So with Chian in the lab, we tested this for COVID. And we were able, in less than 2.5 weeks, to develop this assay for COVID. And you could see he's using serum from people positive. We have a very strong response. And we wanted to use that in a study of case control, where we had at the beginning, it was mentioned by Dell, in January, we had people and they were isolated. We could also look at the close contact, and we wanted to evaluate with NPHL, National Public Health Laboratory, if these people who were exposed to infected patients were also possibly infected. And so we compared different assays. We had beginning of RDT at that time. We had the CPAS developed by Professor Wang Lingfa and Tan Chiwa. And we realized that, in fact, our system was much more sensitive in detecting people who have been in contact with the virus, and sensitivity really was 97%, even in asymptomatic infection. So this technique was very powerful for doing surveillance. So we move on, and I would say we use this technique for many different aspects during COVID. for disease management, for vaccination, for following infection, for looking at the efficacy of treatment. And I will start with symptomatic infection. We wanted to see if we could detect very early people who were infected. And in fact, we could also measure rapidly that antibodies were detected as early as 5 days post-infection. And most of the people, in fact, which were infected developed a very strong response by day 21. And because this technique is flexible, we could use a secondary antibody which recognizes different type of isotype. We could measure IgM, IgA, IgE isotype for 1 to 4. And we, by analyzing the data, we realized that if we had pulling together IgG1 plus IgG3 and divided by the same number of IgG2 by IgG4, we could see that, in fact, people who were mounting a very high IgG1, IgG3 response were the one who had most asymptomatic response. And those who have really a deficit is these two immunoglobulin isotypes were in the severe group. So again, it was for us a way to triage the patient. Those who at this level were really those who were going into severe disease. So we then use this technique for follow the different cohorts that were established by NCID in Singapore when we wanted to test the efficacy of vaccination in our elderly population, because most of the previous vaccination trials were done in people which were between 17 to 50 years old. So, and it was more and more clear that most of the patients and the severe patients were in the elderly groups. So we set up this cohort with NCID and we tested, and it was a multi-groups effort, and we tested our technique in this cohort. So we had the primary regimen which was established at that time, priming at day 0, boosting at day 21, and so we could follow, the patient here. We had something like 300 people that we tested, and we could see that after 21 days we had a very strong response, but not in all participants, and mainly in the elderly. Some didn't respond at all. By 90, most of them had the seroconverted, but the response was not long-lasting. At day 180, a lot of the people had already lost the response. So the peak IgG response was at 90 days. We had a group of door responder which was in the elderly, and really when we plot according to age, you could see that the response, more you get old, the more you have a low response, and the difference was at the 60, which was not a good sign because I just turned 60 at that time. So I thought I fell into that group, which was not for me a very good sign. So age was a risk factor in the vaccination. So that's really, we transmitted this data to the MOH and really encouraged for the boosting. So we boosted our elderly, and you see the boosting was really very good because now all the elderly population, even those with very low response, now mounted a good antibody response. And again, importance of booster vaccination in specific groups. So then we tested for the boosting different vaccine groups. At that time, we had also the coming of bivalent mRNA because we had all the variants and the Novavax vaccine. And you see, in fact, that the boosting at the fourth injection with the monovalent Wuhan strain was the best. Even the bivalent was okay, but not as good as the monovalent. And Nuvaxoid was a bit lower in terms of response. And the advantage also of this technique is that we could clone in all the variants. We have a huge library of all the coronavirus. We could also evaluate the response to the different variants. And again, following the same cohort and using the same approach, because we followed these people for more than 2 years, and we realized that even in some were vaccinated, they have breakthrough infection. And we could really now realize that some people, even if they have breakthrough infection, it was Omicron, the response was, in terms of IgG, was the same. But what was surprising, and in fact, that the IgA level were lower. So in fact, and IgA mainly at the upper airways, so telling us that IgA in the upper airways was really protecting against reinfection, much more than the IgG. So what is the future of S-flow? S-flow was for the spike protein. And so now we move to P-flow for pathogen flow. As I said, this is a very versatile technique. We could put any genes of interest here and have this detection system, really sensitive. We could also clone in smaller fragments, so it's really adaptable if you want. If you have large protein, you could focus on those who may contain the most important B epitopes. As I say, we have developed a panel of SARS-CoV-2 variants. We have also extended to seasonal coronaviruses, flu, dengue, chikungunya. I didn't put the list because now it's large. We also have RSV. And in fact, this technique came from my work on malaria, that we use that to identify vaccine target for vaccine development. And so now we have also refined the plasmid here. It's really plug and play to go faster. So we could produce this plasmid in less than a week. And we could now also use for antibody binding, but this could be also used for other function, and it has been extended to use for ADCC and ADCP with a specific cell. So it's a very versatile system, and I'm happy to collaborate to anyone who is interested with that. So I was mentioned, this really was done during COVID. It involved a lot of people which are in the room here, and during COVID, my lab and Isla's lab fused just to be able to respond to all the multiple cohorts that we have to deal with. And I thank you for your attention. I could take any question if you have. Yes. Antonio.
So, very interesting. How can you be sure that the protein that you want to express in the cells are sticking out from the membrane and not, for example, retained?
Okay, because I didn't have much time, but we have two tags in both sides. So we have a HA tag and a ES tag. And so we used antibody against these two tags to make sure that they are expressed on the surface.
And they're going out. And you never have problem of proteins that are retained and not going out.
Yeah, but they will not show in the flow. Mm. So yes, especially most of the viral protein usually are not very difficult to express. Malaria protein are a nightmare. We are very happy if we succeed for 30% of them, and then we redesign. So there are all the way we could change the codon, make shorter fragment. We always succeed to express.
Thank you.
Thanks, Laurent, over here. So the two questions. The first is, of course, this is only measuring binding, and so have you looked at how this might correlate with neutralization, which is obviously going to be more relevant to protection?
Thank you for this question. We did that. We did in parallel with real viral neutralization assay. We did with CPAS also, and it correlates perfectly. Because in fact, these neutralizing antibody are a fraction of the total antibody pool. But it always correlates.
Okay, great. And then the other question was, you showed that the ratio of IgG1 plus IgG3 over 2 and 4 predicted those people with asymptomatic or mild disease. Now, we know that multiple doses of mRNA, and you didn't show it, but I presume you've also looked at it, induce IgG4. Yeah. So what does that tell us about the consequences or the potential consequences of that IgG4 being induced by mRNA, but not by, say, the protein-based vaccine?
I think the increase of IgG4 is more a regulatory mechanism, because you have this huge inflammation at the beginning when you vaccinate. And I think it's a feedback response, because IgG4 are controlled by more Th2 type cytokines. But these IgG4 are still neutralizing. So it's good that you asked this question. It was in the context of infection that we find this isotype imbalance. In the vaccine context, is a bit different. And in fact, the isotype from the IgG category are less important from the cohort that we followed than the IgA, which is, I think, the clear factor for preventing reinfection.
And with respect to the IgA, so this was serum IgA, just to be clear, you weren't measuring mucosal IgA.
Okay, so it was serum IgA, but we have, again, I have 10 minutes, but we also, this technique we have adapted to the nasal IgA, and we find the same thing.
So the serum and the nasal,
yes, nasal
IgA
and serum
correlates?
Completely correlates.
Okay, excellent, thanks.
Thank you for the talk. My question is a bit follow-up to Antonio's question. So in your system, you showed that you can even express large antigens, but my question is actually the other way. Is it also possible to express parts of antigen or smaller peptides on the surface? Or have you checked that what was the smallest length of antigen which was successfully expressed on the surface to use for this assay?
Okay, that's a good question. We went to 10 kD molecule. I don't think we went below that level, below that size. But I think it's feasible. But after if it's too small, I don't know if it's going to be easy to detect because we have our tags and so on. So the smaller it's going to be hidden in the construct. So I would say that 10K is a limit that I will choose.